Let's say you have an assembly, either generated in Patrick or elsewhere, and you want to annotate it. But when you get to the annotation service, it asks you what taxonomy your organism is. And let's say you don't know. Maybe you're taking a class, and this is an exercise from class that you have to identify this particular genome, or perhaps it's just something that you've been working on and you're not exactly sure what it is. Patrick has a service that can help you with that. So let's go into services and click on Similar Genome Finder. The service uses MASH min-hash, which is described in this publication. But basically what MASH does is it reduces large sets, sequences and sequence sets to small representative sketches from which global mutation distances can be rapidly estimated. Generally in Patrick, we don't want to reinvent the wheel. When there are bioinformatic programs or pipelines that work well and that bacterial researchers are using, we like to bring them into the resource and give those pipelines credit, but also it makes it easier for our users. So we really like MASH min-hash. It works really fast and it's really perfect for this service. So in a previous video, I showed you how you can use a genome. This time we're going to look at a FASTA file. So you could look at one that you had done recently, or you could click on the folder and upload a sequence from your computer if you wanted to, and note it would be tagged correctly. So you could go in and click on select file, and that would give you a direct access to your computer. So I could even choose a folder or a file like that, open it, and start the upload. Once it's uploaded, there it is. I see it here in my file. So I'm ready to figure out who this is. I could search right now, but I just want to point out that you can select the number of hits. You can filter on p-value threshold. You could filter on the MASH min-hash distance. And you could select from the NCBI reference or representative genomes, or look at all the public genomes. But initiating the search is as easy as clicking this button once it turns blue. So we click it and then we wait. Now what's different about Similar Genome Finder from things like our assembly service or our annotation service or any of the myriad services in Patrick is when you launch a job, one of those jobs, you'll see the progress on your jobs page. These are launched on the fly, and they show you the jobs on the fly. And this is kind of the kind of table that returns when you have the information. It tells you the closest hit, it goes from closest to least close under the parameters you selected. It gives you the genome status. It tells you the number of contigs. If the isolation country and host name is available, it'll tell you that, the year it's collected, when it was completed, and then it'll show you the MASH distance, the p-value, and the number of k-mers per 1,000 k-mers. So this looks like a pretty good hit. Note when I click here, it gives me more information about this particular genome along the side and direct links to other sources of that data and information like the biosample, the assembly accession, and the GenBank accession. You can select everything if you're interested, and you could download the table here. You could go to a view of these particular genomes, or you could create a genome group. If you wanted to look more deeply, like at the protein family's presence and absence between this group and your group, you could do this via the protein family sorter tool. So this is a very easy, fast, quick way to determine what the closest genome in Patrick is to yours. And if you're using the annotation system, now I know that I could put in Brucella canis when it asked me for the taxonomy of the organism. In the next video, we'll talk about finding the closest genome using FASTQ files. Thanks.